Um, Thanks to the organizer for giving me the opportunity to talk today. I want to just give you a little touch of our project, and there is a focus on understanding the novel roles of DNMT3a and DNMT3b at the enhancer gene body during human epidermal cell differentiation. The epidermis is a multi-layer epithelium that relies its function on a basal layer of epidermal stem cells that proliferate, migrate, and differentiate upward, forming the different layer of the epidermis itself. It's well known that different epigenetic factors are differentially expressed throughout the different layers of the epidermis. And we wanted to investigate this dynamic expression, and what we could do in our lab is to culture in undifferentiated condition this human epidermal stem cell and trigger the differentiation, adding calcium to the media. And the results that we had from these big experiments that many genes are upregulated and many genes are downregulated. But interestingly, these genes, often belong to the same families of epigenetic factors. Here I give you an example. The DNMT3a is upregulated, instead DNMT3b is downregulated during the epidermal stem cell differentiation. DNMT3a and DNMT3b, just to briefly introduce them, they are the novo methyltransferases, so are the one that establish the DNA methylation in the mammalian embryos. And so to further investigate the activity, the function of our guys in epidermis, we perform chip sequencing of DNMT3a and DNMT3b in both epidermal stem cells and differentiated keratinocytes. And the results that we had, they're like a strong dynamic profile of unique genomic localization, both for DNMT3a and DNMT3b. They can clearly see like over binding of DNMT3a in differentiated keratinocytes that follows the upregulation of the protein, and DNMT3b that is downregulated, there is less binding in the differentiated keratinocytes. But what kind of binding pattern do they show? DNMT3a binds to active transcription start sites. DNMT3b instead shows a large binding domains spanning gene bodies, and both the proteins, DNMT3a and DNMT3b, binds to the most active subset of enhancers interestingly. DNMT3a binds to active enhancers, so like a subset of the binding domains of DNMT3a was found at transcription start sites, and we could see that these transcription start sites are actually active because we have done chip sequencing for H3K43 methylation, histomodification enriched in active transcription start site. And interestingly, the gene expression of the genes they are bound by DNMT3a and they are enriched for H3K43 methylation is higher compared to the one that have just H3K43 methylation. And the second interestingly binding pattern that I want to talk to you today is that DNMT3b binds large domains spanning to gene bodies of active genes. Here I give you an example of essential gene for epidermal stem cells. There is keratin 5 that is highly expressed, that is highly bound by DNMT3b in epidermal stem cells. And this binding is actually very dynamic. In fact, when you differentiate the cells, DNMT3b binds essential gene for differentiated keratinocytes, that in this case is involucrin. And if you do gene ontology of these genes that are bound uniquely at epidermal stem cell by DNMT3b, you have a function of the cell. So you have like keratinocytes, development of epidermis, cell proliferation. Instead, when you differentiate the keratinocytes, DNMT3b binds uniquely to cornified envelope keratinocytes differentiation. So you really give the function of the cells that we are looking. But since there are Methyltransferases, you are maybe guessing, what about DNA methylation at gene bodies of this bound by DNMT3b? Well, we have done MEDIP sequencing analysis in both epidermal stem cell differentiated keratinocytes. We could see there is a large of these genes bound by DNMT3b are enriched for DNA methylation at gene bodies. Both epidermal stem cell and also differentiated keratinocytes. Interestingly, like the gene ontology between the one that contain DNA methylation and the one that do not have DNA methylation, actually very interesting, but I don't have the time to talk to you today. The third binding that I want to talk is that DNMT3a and DNMT3b binds to the most active of enhancers. And to map and find an answer in our cells, we have done chip sequencing for the histone modification related to enhancer domains. There are H3K4 monomethylation, H3K27 acetylation, but also we have done RNA-6 transspecific. You can see the specificity of the RNA, and we enriched for polyadenylated in not polyadenylated, so we could identify the RNA produced from the enhancer domain that are normally not polyadenylated. And here you clearly see like a clear binding of the DNMT3a and also the DNMT3b at this enhancer. The overall DNMT3a binds to 10% of the whole enhancer in our cells, and DNMT3b a little bit less. And here I give you the signal in the enhancer about the DNMT3a, DNMT3b. Interestingly, the mRNA expression of the closest gene in epidermal stem cell and differentiated keratinocytes are like higher expressed both in DNMT3a and in DNMT3b. Compared to the one that do not contain DNMT3a or do not contain DNMT3b. And if you're wondering if the activity of the enhancer is not well measured by the mRNA of the closed gene, you're probably right. And thanks to our bioinformatician, Debayan, we found a very nice way to normalize and to quantify enhancer RNA expression, so we could see that actually the genes that are bound by the enhancer that are bound by the DNMT3b are actually high expressed compared to the one that do not contain the DNMT3b. The DNA methylation enhancer shows that these enhancers are like bound by the DNMT3b are hypomethylated compared to the others. Instead, the ones that contain DNMT3b are like at the higher level of DNA methylation, especially around the enhancers. To understand if the DNMT3 and DNMT3b were important for maintaining the activity of the enhancer, we have done knockdown, a chip sequencing of K27 acetylation in knockdown cells. And the K27 acetylation does not really change between the wild type and knockdown cells. But if you look at the enhancer RNA expression that we've done still with RNA sequencing, we could see that the typical enhancer downregulates the eRNA expression in absence of DNMT3a and also DNMT3b. The conclusion I want to give you today is that DNMT3a binds active and transcriptional cell sites, DNMT3b to very broad domain spanning gene bodies, and DNMT3a binds to the most active subset of enhancers. And a high percentage of these active enhancers are actually super enhancers, something that I couldn't talk to today, but it looks very interesting because it dictates cellular traits and cell identity. And knockdown of DNMT3a and DNMT3b looks decreasing activity of these enhancers. I want to finish thanking the lab. Especially I need to thank Salva for the great support, for always help, and all the lab, Gio, Alex, everybody is just amazing. The lab is great working with them. And it's special thanks for the Bayan, our bioinformatician, that has a great patient with me, but I think we're a good team together. And all the core facility, obviously, like also the CRG lab, or Luciano, Luis, they helped me a lot in the project. And thanks for your attention.